Welcome to the summary video of the activity and temperature chapter of the first module. In this video we're going to go over all the 12 content dot points and pracs and cover them relatively quickly. So if you happen to get lost on any of this summary part, I'm always going to say the video that was the dot point that we're covering. So if you have any problems, just look at the video again and you should be fine. But hopefully the extra summary will help. If you haven't looked at the videos, I would recommend to look at the videos before you look at the summary slide. That will help, but even if you don't, you should still be okay and you should still be able to learn something from the summary slide. Alright, so first video we talked about the role of the enzyme, and this was broken into 3 different parts. First part of the syllabus dot point says, identify the role of the enzymes in metabolism. Remember that word metabolism was just all the chemical reactions that occur in the body. So identify the role of the enzymes in chemical reactions that occur in our body. That's what that part says. And the role they play is they help speed up chemical reactions. So remember, this is here the enzyme. This here is the active site. That's where the substrate binds. And this is the thing that's being broken down. And without the enzyme, this substrate won't get broken down. Or it will get broken down, but it'll take a lot longer than without the enzyme. So what the enzyme does is makes these new molecules from the old one, which we call the product. So the product. So enzyme speeds up chemical reactions, and remember that was a catalyst. And it's not consumed itself. So we can use enzymes over and over again. So they break down these chemical substances into new substances, and they can do this over and over again. So that was the role of enzymes. Describe the chemical composition. So what they were proteins. So this here is a protein, and proteins are made up of amino acids. So each of these dot points, little rings here, is an amino acid. So basically you have proteins which are made up of lots of amino acids, and that makes a protein. And all enzymes are proteins. So their chemical composition is that they're proteins, and proteins are made up of amino acids. Third point of that dot point was use a simple model to describe their specificity. Remember the lock and key? The lock and key was that you had that perfect fit between substrate and enzyme. So the substrate fits in perfectly. And then the substrate gets broken. So they form a enzyme substrate complex when this happens. And then the substrate gets broken down into 2 products. And that would be the lock and key. That the enzyme might be the lock and the substrate the key. Perfect fit. That was one name of one of the models. And the other one was the induced fit. And the induced fit is that if you can look at here, you've got this shape being a bit of a rectangle. And here it's not a pure rectangle, it's a bit of more curvature in the enzyme. So it's not a perfect fit, but it's almost perfect fit. But what the substrate does is when it binds, it forces a change in the shape of the enzyme. And then it works, the enzyme works and breaks it apart. And then the shape of the enzyme goes back to normal. So the enzyme is not changed in its shape, only for a split second whilst it's breaking down this new enzyme, this new substrate into a product. And remember the specificity, there's specificity, so all enzymes only work on one type of substrate. They don't work on any other substrate. So for example, lactase, the enzyme lactase, only works on the molecule lactose. No other molecule will be worked on by that specific enzyme. Right, so we also have the pH. Identify the pH as a way of describing the acidity of a substance. So remember, this is just general. This is knowledge-wise. You don't need to know, but the more hydrogen ions, the more of these there are, the higher the pH. So if you've got lots of these, that means you have a low pH. Whereas if you have only very few of these hydrogen ions, that means you have a high pH. And remember stuff like that the pH of 7 was neutral. That's the stuff you should know, that the content. That pH of 7 was neutral. pH of more than 7 was basic or alkaline. They're both the same thing, basic or alkaline. And the higher the number, the more basic it is. So if pH of 13 is more basic than the pH of 9. And if you go the other way, so if you go less than 7, the pH of 4, for example, or pH of 1, they're acidic. And pH of 1 is more acidic than the pH of 4. So the smaller the number, the more acidic. Whereas if you increase from pH of 7, the higher the number, the more basic it becomes. And remember, we need to know that because either at too low or at too high pH will both denature our enzyme. So we want to keep it at that optimum pH. Usually in most cases in our body it's a pH of 7. If we go too low or too high, it will denature enzymes, and then it will decrease our metabolic efficiency. We also did this prac. So remember the prac, we had the enzyme, so the actual syllabus dot point said we should test the effects of increased temperature, change in pH, and change in substrate concentration on the activity of a named enzyme. So again, this is the enzyme that we use in this video, was the enzyme renin. And what it does, it turns normal milk into clumpy milk. So basically we know that the enzyme was active, that something happened if normal milk turns into clumpy milk. That's our test. So we've tested, like for example, if we looked at pH, if we tested for pH, what we would have done is we would have kept the temperature same, don't change temperature. Only thing we changed is the pH. So pH of 3, 7, 10, 3, 7, 10, right? So we wanted to see what the pH does to the activity of the enzyme, so how fast the enzyme works. And what we would have found is we would have had no change for all the substrate ones, because there's only a substrate here, S for substrate. The enzyme was not in the actual test tube, which means enzymes weren't there, so chemical reactions weren't happening, which means that the liquid milk didn't turn into clumpy milk. We would have noticed that a pH of 3 and a pH of 10 didn't really give a result, because that was not the optimum pH for the enzyme. And the best result would have been roughly a pH of 7, because that's where the enzyme could do its job the best. That's where it had the highest activity. That's what we're testing for, how fast the enzyme works, and it worked best at a pH of 7. That was its optimum pH. Right, so these graphs is what you would have looked at. So this has activity on one side, pH on the other side. For the enzyme, so enzyme activity, it will be high here, medium here, low here. And then we've got pH 1 to 10. And its optimum place spot, which would be right here, is at that pH of 7. So if we go either lower or higher in terms of pH, our activity of the enzyme drops down. It becomes less active because enzymes become denatured. It doesn't always have to be 7, but in this case, this enzyme works best at a pH of 7. Same thing, activity of temperature. So if we check for temperature, what we would have found is that the activity would have been best for this specific enzyme at about 37 degrees Celsius. That's right here. So this would be where it's at highest, its highest point. And if we decrease our temperature or increase our temperature, then we will decrease our activity as well. Right? So optimum temperature for this enzyme was about 37 degrees Celsius, which is also roughly body temperature. And then we also looked at concentration as well, the effective concentration. So concentration refers to how much of something there is in there. So again, we've got on one side, we've got the enzyme activity being low, medium, or high. On the other side, we've got our concentration being either low, medium, or high. And what we actually would have found is that we would have, as we increased our concentration, our enzyme activity also increased, but until a certain point, until sort of we've gone past that medium stage of concentration, then it would have plateaued. And that means that even though we increase our concentration, our enzyme activity doesn't increase anymore. It's going to stay the same. And remember the reason why is because, for example, if you have low enzyme activity, then we have one substrate. Maybe this is just an example. Just imagine this: one substrate and 3 enzymes, which means we can keep one busy, but the other 2 wouldn't be busy. Then we might be increasing this to 3, and then now we keep them all busy. But if we increase it even more, then they're already busy. So even increasing them even more, it doesn't really make a difference. But yeah, if you want to recap that, that's video 3 for this. Next one was, why is it important to keep a constant internal environment for optimal metabolic efficiency? Remember the word metabolic efficiency. So this refers to having a really high, fast speed of chemical reaction. So how fast the enzymes work. High optimal metabolic efficiency means that the enzymes work really fast. Enzyme activity is at a really high level. That's what that means. So again, you can imagine this here is the optimum pH graph we had in the last video. And for most enzymes, quite a few enzymes, they have that optimum pH of about 7 here, right? So that's the optimum pH. So its highest activity is about 7. But what you can imagine, if, for example, you went and had 2 different, a non-constant and a constant internal environment, so that white is the non-constant and the blue is the constant. Just imagine that you woke up and your pH in your body was about 1. Then as you increased your time, so in lunch you got to about pH of 7, and then at late afternoon you had a pH of about 14 in your body. But what would happen to your enzymes? Your enzymes would work the best here. They wouldn't work here at all, or here at all, and they would work bad in between as well. So all of these areas in between, they would still work quite bad. The problem is, if our enzymes work bad, for example, our enzymes in cellular respiration, if they don't work properly, then we die. So if this happens to us, we die. We literally die from this. So that's not good. We want to keep a constant internal environment. So what happens instead is this happens, the whole blue arrow. And that means it's always going to try to stay at 7, because if it goes too high or too low, it's not going to work properly. So instead of having it up or down, it's going to try to keep it at the one optimum area. For pH, that would be about 7 itself. Then we have, this one says, describe homeostasis as a process by which organisms maintain a relatively stable internal environment. So remember, this is, I don't think I've used this example before, but you can imagine if someone said, try to stick to 50 kilometers per hour, that's the speed limit, and you were to actually try to do that, you would try to stick to 50 kilometers per hour. But what actually happened is, so this is a speedometer. I drew a speedometer, and I have that picture next to it to make sure that anyone who's confused about what that is meant to be, that's meant to be a speedometer. But you can imagine it's ideal, but obviously if you always stay at 50, exactly at 50, right? That's a yellow, that's ideal. But if you're driving, that's rarely the case. You're always going to go a bit over, a bit above, a bit under, a bit above. So this blue arrow is at 40, and that red arrow is at 60, or that pink arrow. But in either case, we want to try to bring it back to, so if you're at 40, bring it back to 50. And if we're at 60, bring it back to 50 as well. Right, so we want to keep it at 50, but we're not always going to be at 50. We're going to go too low or too high. So what can we do to try to, if we're at 40, what can we do to bring it back to 50? If we're at too fast, if we're moving too fast, we can obviously go on and press the brake. If we're moving too slow, we can go and go on the gas itself. Right? So if we're at 40, we can press on the gas, which brings us back to 50. If we're at 60, we can go on the brake, and we go back to 50. So that's an example of homeostasis. Homeostasis is about keeping a constant internal environment, but also of how the body does that. So we don't brake or gas. We don't have brake or gas in our body, but we have different mechanisms. So for example, this graph here shows if you're shivering, again, you can imagine this green line to be the ideal level for body temperature. So that might be 37 degrees Celsius. But our body will try to keep that, but it'll always go a bit above or a bit below. But then there's a response that happens when we are a bit low, and a response that happens when we're a bit too high. So for example, if our body temperature just drops a bit too low, like it would have here, and we start shivering. Shivering creates heat, so we go back up. If we maybe have if we're doing too much exercise, and it's gone maybe a bit above 37, we'll sweat, and that sweat brings it back down again. Right? So those two are the same as our brake and our acceleration. The responses that the body has to keep our internal environment constant. So hopefully that analogy made a bit of sense. And the last one here in this first part is explain that homeostasis consists of two stages: detection and a response. So detect change from counter stable state and counteracting change from stable state. So if you imagine in that speedometer example, if we look at if we have at 40 kilometers per hour, what has to happen for us to go back to 50? Well, first we actually have to see that we're going too fast. So the detection stage refers to our eyes actually seeing, our eyes detecting that change. That's the first thing that has to happen. Otherwise, we won't be bringing that back to 50 if we don't see it. The second part is that our eyes see it. That message gets sent to our brain, and our brain tells our legs, our effector, to go on the brake or go on the gas, depending on if you're going too fast or too slow, right? So the leg would be the effector. So leg would be the muscle or the organ that's having an effect, that's trying to bring it back down or back up. And then the response itself is, if we're going too fast, the response will be a brake, and if we're going too slow, then the response will be the gas, like pedal on the gas, right? So know what detection and response, what that refers to. So we have to detect the change from stable state, and then we have to respond to it. And usually there's an effect in between, which is the actual organ or muscle that makes that response happen. And an example in our body would be if we have, this is the blood vessel here, if we have heat swimming in the blood vessel, and the heat might be at 38 degrees Celsius, that was our stimulus, because stimulus is anything that is not normal or away from the normal state. And remember, it's supposed to be at 37, so 38 is not normal. So that heat particle in the actual blood will get picked up by a receptor. Receptor detects that change, and then from there, the detection part, the receptor in our blood vessel, that gets sent to the brain. Brain is our control center. It decides what to do. The brain will send that message on in that yellow form and go to the sweat gland. A sweat gland is our effector because the sweat gland is going to make that response happen. Sweat gland is a receptor, and then the response itself is sweating. Right? So we've got our detection, our receptors in our blood vessels detecting that change. We've got our sweat gland being the effector, being the one that will make the response happen. And then the sweat is the response itself to bring it back down to normal again. Right? So hopefully that was useful. We're going to have the second part in a second as well.